Welcome everyone to this lunch seminar. It's my great pleasure to welcome Seppe Kuehn from Chicago University. So Seppe did his PhD at Cornell University, which is upstate New York, right? Syracuse, which is already Ithaca, which is already one of the world famous, I think, physics departments thinking about complex systems, networks, and also about statistical learning, of course. And then Seppe moved to New York City, so which is a little bit, I don't know how long.
5 hours.
5 hours by car and 10 hours by train, probably.
Not possible by train.
And to the Rockefeller University in New York City, where he worked in the lab for living matter, I think, with Stan Leitler, on different problems already involving, I think, evolution.
Ecology.
And ecology. And then he moved on to Urbana-Champaign, which is in the middle of cornfields. I would say because I took one of the train ride from Chicago down to Urbana-Champaign, and it's just cornfields for 5 hours train ride. 2 hours. And then he recently, or I think a few years ago, moved to Chicago University. So it's a great pleasure to have you here, and we are very curious about learning more lessons in statistical learning.
It's good to be here. I've given a lot of talks in the Netherlands in the last 6 months because I've been on sabbatical. So I'm trying to not give the same talk too many times because I'm worried the audience will have the same people in it. So I'm diversifying a little bit, and this is a talk where I'm going to use the problem of microbial ecology from somewhat of an engineering perspective as a platform on which to make a few points about sort of what it means to understand a system and what is the role of statistical learning in understanding those systems. I have some relatively strong opinions on this, so I hope to elicit some responses. But let me contextualize the problem in terms of the scientific problem that I focused my entire lab and really the last 10 years of my career on. And that is, the problem that I'm interested in understanding and really and ultimately solving is decoding the functional properties of microbiomes. So if you're not an ecologist or a biologist, you might not think of microbiomes, which are complex microbial ecosystems, as functional systems. But I would argue that in everyday life, it turns out that their function is actually quite obvious. So every one of our human guts is now working on one of those sandwiches. And those communities are part of the chemical breakdown of that sandwich, and also of regulating our immune system and maintaining host health, inhibiting pathogen invasion, and so on. This gut community is very complicated. I'll get into the complexities of that in a moment. A bit more central to my own interests is the functional properties of microbiomes in natural environments where they play an important role in things like climate change. So I'll talk only a little bit about it today, but the process on the right is called denitrification. It is the process by which nitrogen goes from a sort of soluble form in the ground up into the atmosphere. And this is a very important aspect of the nitrogen cycle. And it's done exclusively by bacteria in soils. So the question is, we want to understand these functional properties of these systems. These biological systems, like all biological systems, they actually do something. And I think that is a very important thing to remember, especially for physicists like me who've transitioned into these fields. So since I assume the audience is relatively broad, I just want to mention the scale of complexity in these systems is a little bit hard to imagine from where we stand here in the macroscopic world. If I look at a gram of soil, I'm contending with something like 10 million individual bacterial cells. I'm going to ignore the fungi for now because I actually don't know much about them. Those are comprising something like 1,000 species in a single gram. 100,000 different genes are present, and 1 billion viruses that infect those bacteria are present per gram of soil. Those are a high-dimensional system. If you look in the gut, the number of cells in the gut is of order 10 to the 13. People love to say it's the same as the number of cells in the human body. This is roughly true to within an order of magnitude, but the cells in the human body are obviously much bigger, which is why we don't look like bacteria. Again, thousands of species are in the system and they're interacting. And so the question is, how does this really complicated system give rise to these functional properties? And so the problem, as I'm articulating it, is I have some community. It has some set of interactions. Those interactions are taking place between a really diverse constituent of components, taxa, genes, and so on. And out of all of that comes this sort of functional property of the system, this flux of metabolites, the pathogen inhibition, whatever it is. And so what we're really asking is, how can we relate the structure of the system to the function of the system, given this taxonomic structural diversity, the complex dynamics, and the functional properties of interest. So I think with any problem, it's often good to have a strategy. And so the strategy that we've developed after, I will admit, several years of kind of not knowing what we were doing, at least I didn't know what we were doing in my lab. We've sort of developed a strategy which is I feel starting to get maybe a little bit towards the essence of this problem. The first sort of pillar of the strategy, which I'm going to focus on today, is statistical learning. And by this, I mean using statistical approaches to try to glean what are the salient features out of all of this complexity, which we can now measure using sequencing in this case. The second component is, which you will see in some of the work today, is really physics motivated. Write down the simplest model you can for the dynamics of the system, and then somehow use that as a lever to pull on to try to understand the complexity. Do phenomenology in the language of physics. Was a very bad word in biology for many decades, actually. I was advised by Stan at Rockefeller, never say phenomenology to a biologist, because they hear you don't understand mechanism. But I will call it phenomenology because that is what it is. And then ultimately, the thing that we're interested in, we must quantify. So that is, if I want to explain this flux of metabolites through a community, I had better be able to measure that thing quantitatively. And so that is the third sort of pillar of the approach that I'm going to show you. So I want to take you through several studies. I'm going to sort of wrap them together a little bit, but they're in sort of two compartments. The first study, well, there's really two studies, is to relate the composition of the genes that each organism in a community possesses to their traits of those organisms. So those organisms behave in some way in the environment, they eat some types of food, they do it at certain rates and so on. Can we map the genes to those traits? Yes or no? So that's sort of two studies that we've worked on in the last few years. And then the second thread of work is to look at how we can relate the composition of the community. So that is, I have some ecosystem, and I want to understand how changing who is in that ecosystem ultimately affects the functional properties of that system. So these are both really sort of structure to function type questions. You can imagine the structure of the genome affecting the traits of an organism, the structure of an ecosystem affecting the function of that ecosystem. There's a strong analogy here to a protein problem, right? Think about protein biophysics. You have some primary sequence of a protein of amino acids. You want to know what reaction that protein catalyzes. Conceptually identical, I would argue. Although the systems are clearly very, very different. We're going to learn a few things on the path here. The first thing, which I will show you, I hope very clearly, and is, I think, especially important in the current moment, that prediction and understanding are not the same thing. And so I will show you situations where I can make incredible predictions and I don't understand anything. And so I think that's important to keep in mind. In some cases, it's okay, you just want to make a prediction, great. But I think we need to be very crisp about what we mean by understanding and what we mean by making predictions. And the second lesson, which I think is also important in general when we're studying complex systems, is that sometimes less is more. When we're doing when we're approaching problems that appear to be high complexity, there's a desire often to go to the most complex model and figure out how to use it, and then make some crazy prediction from our data. I'll show you in this context that sometimes trying the dumbest thing first isn't really the worst idea ever. So the first study is to try to predict metabolic traits from the genes that organisms possess. This was done by a former postdoc, Karna Gowda. And the idea is to ask, how does natural variation in the genomic structure of an organism affect its metabolic capabilities. We're not the first people to try to attack this problem, but it turns out to be a pretty hard one. So we want to make this prediction. Sorry, bacterial genomes are not always, but mostly circular. So I've drawn them as circles with little blocks that are themselves supposed to be the genes. How do those genes affect the traits of those organisms? We're going to be working in a metabolic process, which I now know and love quite well, but I'll just give you a brief overview. It's called denitrification. It is this sort of cascade of reactions from nitrate on the left to N2 on the right. And different organisms can perform different steps. And thanks to bacterial geneticists for 40 years or so, we know the molecular components in the cell that are necessary to perform that process. We don't know biophysically exactly how they all interact quantitatively, of course, to make these predictions. So here is the approach that Karna and an undergrad in physics at Urbana, Derek Ping, took. What they did was they went to the cornfields in Urbana, they isolated a bunch of strains, and they phenotyped them in the lab. So they made these painstaking, this is the training data effect. They made these painstaking measurements of essentially the dynamics of each one of these strains performing each step in this metabolic cascade that they're capable of. So how fast do you do the first reaction? How fast do you do the second reaction? How much biomass, how many cells can you generate per unit nutrient for the first reaction and the second reaction? That was all measured. And what they did was they fit a model to the dynamics of these metabolites, and that model gave us parameters like RA and RI, which correspond to these metabolic traits. So now we have a library of organisms where we've quantified the metabolic traits, and the game we're going to play is to try to connect those to the genotypes. So as I mentioned, we know the genetic sort of players in this pathway. We know which genes, their names, what they look like in sequence space, and so on. We know those, and we can annotate them in these genomes. And so that returns for us, after we run the sequencer, a matrix which has as rows species and as columns these genes, and each dot is, yes, that species has that gene binary variable. And these genes are sort of classified into different types of enzymes. Some are enzymes that perform this reaction, some are regulators at the regulatory level that affect their expression and so on. We are leaning on a very important feature of bacterial genomes, which is not a feature of your genome and my genome, and that is use it or lose it. If you look at a bacterial genome, there's a couple of very important features. Number one, there is no extra space. Everything in the genome is coding for a protein. Not everything. 95% of the nucleotides are involved in protein coding. So that means if there's a gene in there, it is typically doing something. And if it's not doing something, the selection pressure to get rid of it is very strong. And so you rarely see genes that are not actually coding the functional enzyme. That means that these zeros and ones mean something to the organism. If these were eukaryotes, especially larger metazoan eukaryotes, this would not be true because their genomes are often littered with stuff, viruses. But what that means is that I can take the binary vector of zeros and ones and write that down and take it seriously as a genotype. And then I can ask, statistically, can I just predict the phenotypic trait from the binary zeros and ones, the genotype? And each coefficient in this regression is then the contribution of having that gene to that trait. So for example, a large positive coefficient would mean that if you have a 1 in position J, you get a large boost in the rate at which you perform this first step in the reaction. So remember, this is a measured quantity. This response variable is a measured quantity that we measure strain by strain in the lab. Of course, we have to regularize. I don't want to talk about it, but it's a necessity here. So what's remarkable about this is that it works. What I mean by works is what I'm showing here on the left. So I'm plotting on the x-axis the observed rate of each strain performing the second step in the cascade versus the predicted rate, and each point is a strain. And we can do some careful holdout data set analysis to show that these predictions are not derived from overfitting. And the other thing we get then is a series of coefficients which tell us how important for this prediction each one of these genes is. And it turns out that some of these actually are sort of mechanistically interpretable in terms of what we know about that. In particular, we know that there are two enzymes that perform this second step. In our regression, this NERK enzyme gets a large positive coefficient. And if you go look in the literature, that enzyme is known to be faster in vitro and in vivo. And so it sort of accords with what we already knew biologically about the pathway. But keep in mind, we didn't tell the regression about this. It's just kind of popping out in our lab after the fact. Okay, so this, I think when we wrote this paper, I think it was an important contribution because it showed that this statistical approach can sort of work. The downside, in a certain sense, and one of the critiques we got, which I think is justified, is that this is a simple metabolic process. If you're a metabolism wizard, you would say, oh well, it's just an electron acceptor. What about all the you can say all sorts of things. Maybe this statistical approach doesn't work if you're dealing with a genetically encoded process that involves 500 enzymes or something, right? Something much more complicated. And so in that case, maybe your statistical approach isn't so well justified. So why not try this for complex metabolism? And part of the reason this is a salient question is that, of course, there are methods to try to take the genotype of an organism and predict its metabolic phenotypes from that genotype. We're not the first people to try to solve this problem. The problem is complicated. Why? Because the metabolic networks, of course, that give rise to these metabolic traits are themselves really complicated, like the one I'm showing here. And I just want to give you sort of a bird's eye view of metabolism, the physicist rendition of metabolism. You take some nutrient in on the left, let's say it's a carbon substrate. That carbon substrate is then catabolized, broken down into constituent components. This all happens in this network, which has at its center glycolysis and the TCA cycle. And then the output of this network is anabolism, which is the process of building cells out of everything you broke down from the catabolite. Okay. And that's what this whole complex machine does. So the prediction that we wanted to make, Zechen and Ahmed and I, was I give you a carbon compound and a genome, and you tell me, can that genome produce biomass, yes or no, up or down? That's the problem we want to solve. And so there's an existing paradigm for this, which is, it turns out quite dominant, especially when one submits grants on statistical approaches. The first question is, we already solved this problem. And so I'm going to argue that we didn't actually solve this problem yet, and I'll show you what I mean by that. We solved it in certain contexts. So this approach works very well for model organisms, but if I go pull a bacteria out of the ground, which has some complex genotype, which I haven't seen before, this approach that I'm about to outline, I'll show you in a moment, really doesn't work. The idea is the following. Go to the soil, isolate an organism, sequence the genome, annotate all the genes, say which genes are there, name them, gene 1, gene 2, gene 3, and so on. From that, build a network, a metabolic network from all of the vast knowledge we have of enzymes, then simulate that network at steady state, the whole network, every reaction, just under the assumption of steady state. And then use some constraints to find a unique solution to that problem, which turns out to be a little bit hard. And from that, make a prediction. That's how it works. And so what you can do then is you can go from literally from a genome with a piece of software that people have developed to a metabolic network, to a prediction. I give you glucose. Yes or no, you produce biomass. You're able to grow. Which means, what does it mean? It means you're able to catabolize that to produce all of the precursors that you need to make a cell. Protein, lipid, blah, blah, blah. Okay? Okay. So does it work? So this is the experiment that we did. This is an experiment designed for a theorist, which is what Zechen is. It's very easy. You take a bunch of organisms that you isolated out of the ground, and for each one of those organisms, you grow them on many different carbon sources. So I give them glucose, I give them succinate, I give them glycerol. I choose these carbon sources somewhat intelligently. And then I measure, can you grow, yes or no, up or down? I just measure it. And so what I get then is a matrix with 100 rows, which are strains, and 10 columns, which are different resources they could use to build cells. And each entry of the matrix is telling me, yes, you succeeded in building biomass. No, you didn't. Okay, those are the two outcomes. And then what I do is I sequence all of those genomes again. And I have another table now, which is genes by strains. Okay, again, another matrix. Great. Okay, so now what I can do is I can go back to this flux balance analysis approach, and I can turn the crank. So what I'm going to do is I'm going to approach this problem two ways. One is to make predictions of the growth phenotypes from this automated metabolic model generation, and another to try to make these predictions by regression. Okay, so this figure is a little bit busy. I'm sorry for that, but let me just walk you through it and make sense of it. So each one of those columns is a set of binary random variables, which is telling me, did you grow or not grow on this carbon source? Okay, and each one of these rows is a different carbon source. And so I can imagine the probability of randomly getting the right phenotype just from a Bernoulli random variable with the same probability of ones in each column. So I can create that null where I just shuffle randomly. And I say, what's the probability I get your phenotype right if I just guess randomly with the right probability of ones in that column? That is essentially this dark gray violin plot. So these dark gray violin plots are all of those guesses, okay? The black X is the guess from the automated construction of the metabolic model. So what that's telling you is that when I construct a metabolic model from the genomes, I'm not doing any better than if I flipped a coin to guess the phenotype of that organism, whether it can grow or not grow on that carbon source. Okay, that's bad, I would argue. And I'm currently in a little bit of a fight with people who are using these methods to, for example, design dietary interventions on your gut microbiome. I don't think it works. This is my statement. Now what I can do is I can take all of those genotypes, those 7,000 by 1 vectors, and I can build a statistical model which predicts from that vector whether you grow or don't grow on that carbon source, just by regression. And here I begin to celebrate, right? Here are my accuracies, and of course one can measure balanced accuracy and something more rigorous and so on. My accuracies are really high, 0.95. I'm getting it right almost all the time, out of sample. These are held out test sets. Okay, wow, amazing, I'm so excited, I did so well. Don't celebrate too early is the message here. So why are those, the next question you should ask yourself is, why is my statistical model working? And here's why it's working. So these organisms are evolved objects. They live on a tree. Like they're all related, just like we're all related through some phylogenetic tree in the evolutionary past, right? So there's some tree which relates each organism to each other organism. And what I did when I did this statistical prediction was I held out individuals randomly along that tree. Okay, so I held out the 2 red dots. I trained my statistical model on the blue dots, and then I predicted the red dots, and I celebrated I was successful. But what happened was the model, if you look at it carefully, all it's doing is it's finding the nearest neighbor on the tree and just using that as the prediction. And that's pretty good, because it turns out that if you're closely related to an organism, you tend to have the same trait. So the model is just learning the tree. Maybe you care, maybe you don't care. If you just want predictions, great, it could work. Actually, it works better than the FBA model to just learn the tree. But it means that if you have a test strain that's some way out here on the tree, you shouldn't expect it to perform very well. You can show that it's doing this in many ways, but the most obvious way is to construct your test set in a way that's aware of the structure of the data. So I can instead say, okay, well, I'm going to hold out strains that are closely related on this evolutionary tree, but they're not related to the training data in blue. Okay? And now my predictions are bad, like on par with my random guesses. Okay, so I killed my statistics. Always good. Okay, and the reason that this is a problem, so we spent an inordinate amount of time studying this. Maybe we're just slow, I think. But the reason that this is such a hard problem is that there's just an astronomical number of good prediction distracting features in these genotypes. You have a dimensionality problem. You have 100 data points, 7,000 genes, and many of those genes have very high correlations with the traits. So for example, imagine in this fake data set, these two grow, these two don't grow, these two do grow. There's a whole bunch of genes that will all be equally good in terms of their ability to correlate with that trait. And there are many of them by random chance that will have that correlation structure. But depending on which one you pick, some of them might be good or bad in terms of their ability to predict your test set. And so the true gene, the one that's like mechanistically actually involved in the process that you're interested in, is impossible to find in this ocean of features. And so you have to find another way. You can't do this. Neural network's not going to save you now. So one solution to this is to just get rid of is to reduce the dimensionality of your prediction space, to find the set of genes, a smaller set of genes that you can use to make these predictions. This is always a good idea, if at all possible. Throw out as many as you can. And so what we did was we then used sort of biological knowledge to say, okay, I'm going to look at known pathways that link that metabolite to central metabolism. I'm going to look at pathways that include the carbon source or enzymes that act on that carbon source. And then for some of these, I start to do a little bit better. Okay, you can see that for like arabinose or butyrate or propionate, my predictions start to improve. There's no silver bullet here, though. It seems like this catabolic problem is significantly harder than the first problem that I showed you. And you really have to leverage some underlying biological knowledge to start to win. But this certainly improves things and still definitely beats the full-scale metabolic model. These are the examples where we see this improvement. The second solution, which is a hard one to say to the experimentalist, but I'm the experimentalist here as well, so I can say it to myself, I guess, is go get more data. Your data is not good enough. So your problem needs more data. And so here's one example where we were actually able to find a data set where a bunch of these strains were grown on a single amino acid. And what we can do is we can do a regression. When you have that many data points, 1,000 data points, then these distracting features will start to get reduced in quantity because the number of independent genes that are correlated with your trait will go down just by random chance. And so now what you find is that the regression starts doing really well out of clade when I structure my test set by organisms that are not closely related to my training set. And what's really remarkable now is actually I start learning things that I can propose are mechanistically related. Okay, because ultimately the statistical model has to teach you some biology to be worth it. And so I can look at the genes that are scoring highly in this. This is a random forest regression. So these are important scores. I can look at these genes that are scoring highly, and it's actually quite interesting. The first gene was known. It acts directly on this amino acid and cleaves it. Okay, that's good. I mean, our regression found that. We didn't tell it about that gene. The second gene, you start digging through the literature, you find a paper in the late 90s from these guys, which says this is related to some sodium. Oh, but for some, buried in the abstract of this paper, for some reason, it seems to help them grow on tryptophan, which is this amino acid. So one observation in the 90s in E. coli accords with what we're learning statistically from this regression. So the point is, if you have enough data, you can start to learn features of these high-dimensional genotypes and suggest experiments, I think, even, which are saying like, okay, maybe this gene is actually involved, if you're willing and able to collect a sufficiently large data set. My point is the statistical approach, it's not a silver bullet by any stretch, but it can propose mechanistic insights that one can then go test. Okay, so what I want to say from all of this is that, prediction is not the same thing as understanding. I think there's a very clear case there, right? When we're using phylogeny, I don't think we're understanding anything about the molecules, the proteins that are responsible for those observable properties of cells. We're learning something about the phylogenetic tree. In that sense, you can restrict the feature set or just go do more experiments, which are becoming cheaper and cheaper. Yeah.
There might be another option if you want to, for let's say, grant purposes, show that your method works without collecting more data. I guess you can simulate fake data using the mechanistic approach, train your model on it, and show that it gives an equally valid results, then without having to do all the hard work, can show that your method at least is as good as the original.
It's an interesting idea. I suppose then the statistics of the fake data generated by those models has to somehow approximate the true statistics were you to generate the data, which I'm not clear would be the case, but
I don't think it would have the same caveats as the original
Right.
methodology, but at least you've shown that it works in principle.
that's right.
And then you can argue that it's worth collecting more data.
That's
and
right.
spending money.
That's right. I think what we did here was we generated complex rules for mapping genotype to phenotype in silico. And then we just asked, how does the quantity of data that we have from genomes that have the statistics of real genomes enable us to learn those complex without doing the metabolic model at all? This is more of just a pure statistical.
So like power analysis. So,
Yeah, something like this, something that's related to power analysis. But you always have to have something, you have to know something about the underlying distribution of the data when you do power analysis, because if you don't, obviously, it's wrong. Okay, so I'm going to switch to the second part of the talk, and that is to approach this question now of how the composition of a community affects its functional properties. This is work that I've been doing with both collaborators outside the lab at UChicago here in Europe, and various graduate students that are sort of floating around the US. Abby was a student at Yale and just hanging out in Chicago for a little while, and we got to do this paper. The question is as follows. Imagine I have an ecosystem, a community, a microbial community. There's some interaction structure. It has some functional property. This is like a flux of these metabolites through the system. And then I go in and I zap the orange organism and I get rid of it. And the question that I want to answer is, how does that affect the metabolism of the community? Why do I care about this problem? From an engineering point of view, it's very clear cut, and I'll show you examples where we can apply this to applied problems. You know, if I want to engineer a community to produce a byproduct, to alleviate pathogen invasion, which I'll show you in a minute, to degrade an environmental contaminant, this whole endeavor is currently not possible because we don't really understand fundamentally how changing the composition of a community affects its function. So here is the sort of most common way to go about doing this. The most common way is to try to determine the interactions between each component in the system, to write down a dynamical model for how the blue one affects the orange one and so on. This is a very experimentally expensive approach and even computationally difficult. Then I simulate the dynamics. Once the dynamics go to some state that I'm happy with in my experiment or whatever, then I look at the abundances and I say, oh, these are related to the function of the system. Now what I could do if I had that full model is delete an organism, run my simulation again, get my new abundances, and predict my function from that. People have actually done this. It's painstaking for an ecosystem of 4 organisms, right? And what I'm saying is most of the communities that we're interested in, 4 is not a number that's anywhere near bigger. So actually during the pandemic, I had started having discussions with Alvaro Sanchez in Spain, colleagues at UChicago in the medical school and elsewhere, and we started asking ourselves, there's got to be a different way to do this, because if you're looking at the human gut, if you're looking at soil systems, this game is over. And the idea sort of comes from genetics, really. Genetics has this idea of a fitness landscape, which goes back to Sewall Wright, really, in the early, Wright-Fisher dynamics and so on. So Sewall Wright had this notion, which is extremely abstract when you actually think about it for a moment. The notion is that there's some high-dimensional genotype space, and that genotype space encodes these discrete genotypes, which are the presence and absence of different variants of genes, and that genotype then confers the fitness of an organism in some way. And so above this genotype space, there's a landscape, which is how much fitness that genotype has, its growth rate in the sort of evolutionary sense. This abstraction is really kind of crazy when you think about it, right? Because if you change a gene in an organism, that affects all these dynamics in the intervening level, gene expression, development, blah, blah, blah. So the map between the genotype and the fitness is something really complicated, right? So we think, there was an analogy. I wrote a perspective with Alvaro, which is to say, okay, well, let's treat communities in this way, where the space that we're interested in down here is the composition of the community, and the height of these peaks is not fitness now, but it's some functional property of the system that we're interested in. And now I'm going to get rid of all the, again, just like the fitness landscape, I'm going to sweep away all of the details of the interactions in this community, of the interactions between the genes, and I'm just going to relate composition to function statistically. So there are some useful concepts in landscape thinking which we can use to essentially attack this problem. One of the concepts is epistasis. So imagine I have some genotype, little a, little b, and then there's two mutations, big A and big B. And I can measure the effect on fitness of big B by itself and big A by itself in the background of little b and little a. Then I can measure the fitness of big A and big B, and I can ask how much does that depart from an additive expectation? That departure is called epistasis. Okay, this is very analogous to a JIJ in an Ising model. In a community context, it's the same. It's how does the function of the community depend on the presence of the first strain, the presence of the second strain, the presence of both strains, and how does that community function depart from an additive expectation? That's what epistasis is. If I have a low epistasis in my community, so most things are additive, I have a single peak in my landscape, they call this a Mount Fuji, where I just go to the top of that peak very easily. If epistasis is strong, instead I get this very rugged landscape. And there's a whole literature of relating these fitness landscapes to things like spin glasses, which look, of course, more like this. So as epistasis goes down, ruggedness goes down, and presumably predictability goes up. Okay, so we're going to try to use this formalism to try to solve this prediction problem. Solve is a big word, I don't really mean that, to approach this prediction problem. So here's a data set from Clark et al. This is from Ophelia Venturelli, now at Duke. And Ophelia does these absolutely grueling experiments where she has 25 gut commensals and she grows about 600 different combinations of those strains in the lab in anaerobic environments. And then she measures at the end of that growth cycle the amount of butyrate that they produced. Butyrate's important, it turns out to be important for your gut health. So there's 2 to the 25 possible combinations of 25 species. They measured 600. That is 0.002% of all possible combinations. It's like nothing, right? But they report to us this incredible data set, which is a matrix, again, that looks like this. Each row is a community. +1 means that organism was present in the community. -1 means that organism was absent in the community. And for each one of these communities, they measure the amount of butyrate production. And I'm not going to go into this today, but there's sort of very elegant mathematics behind encoding presence and absence as + and -1. This gives you coefficients which can be interpreted as Fourier amplitudes on this high-dimensional hypercube of genotypes. So you can read about it in this beautiful paper by actually a Dutch person who's a good friend of mine, Frank Polwijk. And he basically lays out this formalism very, very clearly. So in this basis, what we can do is we can ask, if I write the function of the community essentially as a Fourier expansion on this landscape, or a Taylor, you can think of it as a Taylor expansion if you want, that's the 0-1 basis, where I have additive contributions of the presence of each species on the function, epistatic contributions, the pairwise interaction between I and J, and so on, all the way up to third order. So the first question we're going to ask is, do we need epistasis? Like, is there epistasis in these community function landscapes? And the sort of short answer is that the epistasis is quite weak in these landscapes that we're measuring. So here what I'm plotting for you is the R² out of sample for predicting butyrate production from species presence absence. And what I see is that it's already about 0.8 when I'm only using the information of the presence and absence of each individual species in the community. Predictions improve marginally when I increase the order. At third order, I still get a little bit of an improvement, but I'm starting to have trouble fitting all of these coefficients. And so just to give you an idea of how good these predictions are, here's a second order model predicting the observed butyrate versus the predicted butyrate, and showing that that correlation is really quite high. So what we can do is this, this model can predict for you, if you build this community, it will produce this much butyrate. The remarkable thing is, in Ophelia's paper, what they do is they actually build this incredible dynamic model of the community, like a lockable Terra model, with all the interactions between all the species, and they make predictions that look like this. Okay, so the point is, the difference between this and this is very small, and the number of parameters is way different. Okay. So this is sort of a little bit emboldening for us, because it makes us think that these landscapes are actually relatively easy to learn, and we can use them to sort of engineer. So here is the fraction of the variance explained by order of the model. So most of it is soaked up in that first additive, the effect of each species on average to the community function, and the epistatic contributions and third order contributions are relatively smaller, although certainly not zero in this case. So we looked at a bunch of different data sets from my lab, from Alvaro's lab, from other labs, and basically the story that I just told you to a first approximation holds for all of those cases. The epistasis is relatively weak, in some cases a little bit higher, but certainly the predictions remain okay. So there's sort of two questions, which I'm going to answer in the last little bit here, which are about whether this has any utility. I sort of motivated this with some applications. Can we actually use this to engineer anything that does anything? And the second thing is, it seems a little not right that there's no or weak epistasis in these landscapes. Is there something that about all of these experiments that we're missing? Is there in fact epistasis in communities that are subjected to, say, different environmental conditions or have different compositions? So I'm just going to tell you briefly two vignettes, which are unpublished. They're still preprints, but they show the applicability, and one of them shows the origins of epistasis in these landscapes. The first vignette is about inhibiting Klebsiella. So Klebsiella in immune-compromised people, or often just sick people, Klebsiella will take over the gut microbiome. This is very bad news for you. Often happens in hospitals. One of the state-of-the-art treatments is a fecal microbial transplant from a relative, and that can work actually remarkably well. The problem is the FDA will never approve it because you can't prepare feces in a reliable way. The second reason it's a problem is that the outcomes are somewhat variable. In some cases, you get septic shock as a response. So the idea, which is really coming from Arjun Raman, who's a proper MD, PhD, and knows what he's talking about in medicine, is can we design a synthetic community that does this, that has a controlled composition that kicks out that? So we took a library of 45 gut commensals from the hospital at UChicago, and we started assembling communities of those commensals and asked in vitro if they could inhibit a pathogen. And so what we did was we inoculated 100 different communities in the presence of the pathogen, and we measure over time the abundance of that pathogen. Notice the y-axis is log 10, so it goes from 10 to the 9 to 10 to the 4. So some of these communities dramatically inhibit the pathogen. They're managing to kill it. Some of them are not managing to kill it. So the composition of the community clearly matters for how well the pathogen does long term. And so here are communities and the log 10 abundance of the pathogen at the end of this incubation. So now what we can do is we can play this game where we try to predict this number from the composition of the community in this same formalism. So some landscape equation. We're actually doing something slightly different here, but I don't want to dive into it too much. So here is the sort of first indicator that these models are generative in the sort of more complex sense used in the current ML lingo. Here is the predicted KP and the measured KP for communities that were not in the original sample. So we fit a landscape equation to the first data. We computationally sample a set of unseen communities from that model, and we ask which ones. We ask for communities that should have high KP load and communities that should have low KP load. Here's the prediction from the original model and the measurement in the lab. So this is telling me that I can take this model seriously, truly out of sample. Right? It's telling me it can learn new communities which have predictable functional properties. In an experiment that struck me as completely insane, to be totally honest, I thought this would never work. What Arjun and his postdoc Rita did was they took this high-performing community and they put it in a mouse that they induced a Klebsiella infection in. So they take a mouse, they inhibit its gut microbiome, they give it a bunch of Klebsiella, it's very sick, and then they do an experiment where they either add the synthetic community, they do a fecal transplant, or they do nothing. And the remarkable thing is the synthetic community works. So here is the days since the mouse got infected with the pathogen Klebsiella. The red line is what happens to the pathogen load when we inoculate the synthetic community. The gray data is when we do a fecal microbial transplant. It's about the same. And then there's some other controls here, including PBS and some other communities that don't work as well. So it seems like, for reasons that we are still investigating, these synthetic communities that were designed in a dish are translating to the in vivo setting. And I will not say right now that I know why this is the case, but it's compelling, I think, that we can use this approach for applications. Okay, I'm going to take 4 minutes for the last example, and then we'll stop. The other situation in which this can be quite useful is when we think about applied problems such as remediation. So remediation is a situation where you have contamination in a site, typically soil, water, whatever. There's a whole industry where people will take that soil, put it in a bioreactor, and run it in a bioreactor for like 2 years to get the contaminant to go away. And basically what's happening is the microbes are eating the contaminant. And there's various sort of voodoo tricks that people use, but what we're saying here is maybe we can use this synthetic community design to add a special community to that remediation context to get rid of whatever toxin is in that environment. And we're going to work with this compound bisphenol A. So BPA is this stuff that was infamous for being in all of our water bottles about 10 years ago that they promptly took out and then replaced with something that has a different name, but basically does the same thing. So BPA can be used by bacteria as a carbon source, and they'll transform it into harmless CO2. Okay, so they'll catabolize it, just like I talked about earlier, through that TCA cycle. They'll generate energy from it, but it's a hard metabolite to use. Most bacteria are not happy using BPA. But it is known that communities can degrade BPA better than individual strains, and this is because it's believed that they actually cooperate in the degradation of different downstream products as that thing is cleaved apart. So we actually had a very hard time finding bacteria that can consume this toxic compound, but it turns out the biggest producer of BPA in the world is in Indiana. So Mahmoud and Jonathan and Kissa drove there one day and went literally, right around the back fence, scooped up the soil, and it was littered with bacteria that can consume BPA. So maybe true in the Netherlands as well. I don't know, but it was a bit daunting to see that succeed. Now what we can do is we can take these synthetic communities and quantify their BPA degradation. So what I'm going to do is I'm going to mix some of those strains together with some BPA, and I'm going to measure the dynamics of them using the toxin. And so here what I can show you is BPA as a function of time. You can see one community degrades it fast and one community degrades it slow. And Mahmoud is going to do this for many, many, many communities. In this case, 70 communities across different concentrations of BPA. And the reason he's going to do different concentrations is because as this BPA concentration rises, the toxicity of the system gets really egregious. Many strains just cannot survive anymore when the concentrations reach 100 or 150 parts per million. So now what we're going to do is this same sort of landscape approach that I talked about, where our response variable is going to be the area, the integral under this BPA degradation curve. I can motivate that in detail later, but basically that integral, the lower that integral, the faster they degrade, the higher that integral, the slower they degrade. And it's a model-free metric. Because of the dynamics in this BPA degradation, we weren't happy trying to use a model of this degradation. So what we can do again is then take this model and start making predictions, predicted AUC versus measured AUC, should say log AUC, for the different concentrations of BPA. And what you can see is that the predictions start quite good on the left and get significantly worse on the right. Still significant here on the right, but certainly not as good. So what is happening here is that as we go from left to right, as this toxicity rises more and more, the epistasis is becoming more pronounced in these communities. And so the punchline, which I'll just summarize for you briefly in a couple of plots, is that this toxicity drives epistatic interactions between members of the community. So if we look at these epistatic coefficients, these are coefficients that are telling me how important is the dual effect of I and J together over and above the effect of I by itself and J by itself. And what you find is that in this high concentration, this distribution of these coefficients is much broader. There's many more coefficients that are larger in the wings. I just want to say one more thing, which I did not put here. I think the sort of real nail in the coffin of this, whether these interactions become more important as toxicity rises, is that if you take individual strains and you ask them to degrade at this highest concentration where the toxicity is the highest, none of them can do it by themselves, but communities can successfully degrade. And so it means that there must be collective effects that are involved in the degradation when the toxicity is high. Again, we asked whether these communities could remediate a contaminated natural environment. And so this is just a quick teaser. We took soil and added BPA. And in one case, we added our synthetic community. And in another case, we didn't add our synthetic community. And then we measured how fast this environmental contaminant could be degraded by these synthetic communities that were added. And what we found was that when we inoculated a community, even into this very complex natural habitat, it helped the system degrade the environmental contaminant more quickly. Okay, I'll stop. I just want to thank the important people here. So Karna Gowda and, oh, Zechen is not here. Okay, Zechen Lee, I'm sorry, his face is not here. We're responsible for the first part. Rita for the mouse experiment, Mahmoud for the BPA experiment I just showed you, Karna also with Abby, who's up there in the corner, for this landscape approach. And these are some very important collaborators of mine, who maybe some of you know, and the funding agencies. So thank you for coming, and I'm happy to chat more.
Thank you very much. Also time for a couple of questions. Any question? Yeah, that.
Yeah, I was wondering that bacteria are known to have different metabolic strategies. Given, yeah, if there are multiple resources present, they will switch on depending on what kind of composition of the resources that they live on. They will switch on their different metabolic strategies.
Correct.
What's that? What's the impact of that on your analysis?
On the first part or the second part? The first part?
Both. I mean, because to me it looked like that you're not really considering this adaptive behavior.
Not really considering this adaptive behavior. It's sort of interesting. Actually, in the first part, there's a sense in which we are, but not on a sort of grossest scale. So what you're saying is like bacteria, for example, they'll do this nitrate respiration I showed at the beginning. They'll only do it when oxygen is gone. If you put oxygen back in the system, they'll start using oxygen instead. We're not describing that. However, when you look at that metabolic cascade I talked about, it turns out some of the strains will work on the first step for a while and not the second step and then switch. And other strains will not. And our model is in fact capturing those differences between strains statistically, but not mechanistically. So maybe in like a small scale, we've taken like one nudge into that, but on a broader scale, where you're right, bacteria have multiple strategies depending on the organism. We're not saying like, oh, you were doing nitrate respiration and next you'll switch to this other thing. Qualitatively different. No, that's right. I think that remains.
But when you make this synthetic community, how do you adjust the ratio between different bacteria? I mean, it could also affect the.
The short answer is we don't, and the abundances are high. And so I expect, so what we're doing is we're initializing the community always with the same initial abundances of each organism. This is actually a very important point. And we're not varying that by a large amount. So what that means is the dynamics are quite, what we see is actually the dynamics are quite deterministic for a given composition. Like we always see the same outcome and the same function. But I think if you took one of those organisms and you dropped their initial abundance by a factor of 1,000, now you might be in a different regime, right? But we're starting with numbers that are like 1 million or 10 million. And so stochastic effects, for example, I think are not important at that level. But at some point, this will stop working. You'll go into some other state, right, where the composition in terms of zeros and ones is the same, but the final function is clearly different.
A related question, what was the size of the syncoms in terms of number of bacteria tested, especially against the Klebsiella
Number of species or,
strains?
oh, so anywhere between 1 and 46 strains with an average richness in the 20s.
So it differed, so, yeah.
Yeah. And there's a weak effect with richness, which is just slightly higher diversity communities being a little bit better, but that doesn't fully explain the result. Like if you do a model based on richness alone, this is a terrible model.
It's also the regression that showed it wasn't necessarily particularly large or small syncom that explained the highest inhibition.
Exactly. No. In fact, in this BPA story, the community of all strains is not the best community. So there's non-trivial factor in that sort of composition of the, right? I think if that were the case, it'd be in a situation where it's like, okay, just put everything in.
Yeah, it's a following question too. So, is there so the diversity you talk about is always based on species, or did you ever try like maybe violence or between the families?
Yeah. No, we haven't tried this, but something that Ophelia is doing is sort of following up on this. Ophelia Venturelli is following up on this by asking if she can inform the regression about the genotype of the organisms, which is maybe a higher bar even than you're asking for, right? So to actually use the model to predict which genotypes you should go and find and add to your community to make the performance.
Yeah.
That's going to require some data to get right. So I'm sorry.
Any more questions? Yeah, they go.
That's one.
You're okay.
In the example of Calypsida, so when you change a different type of community, it goes up and down. And you have also metabolic model and carbon source. Can you play with carbon source instead of adding this community to gut, or is it?
Great idea. So I know it's something that Alvaro is doing, where the design space is the nutrients and not the composition of the community. And I will say that ultimately, I think this will work better. And the reason is that when you have natural communities like human gut, there's actually a very nice paper out of Chicago last week in Nature showing that using diet to manipulate the gut microbiome is far more effective than using synthetic communities. And the reason I think is that when you look at a natural community, it's very hard to invade that thing. It tends to be highly packed in a sort of niche sense. The same is true for soil. If you try to put something in soil and you come back in sequence and look for it 2 weeks later, it's gone. This has been done many times. But what you can do is change the environment, and then everything in that system has to adapt to that environment. And that ultimately might be the better engineering approach to these communities, in my view. I don't know. I think it's an open question, but it's a very astute question.
Okay, let's thank Seppe again.